All right. Hi, everyone. My name is Xiaojun Wu, and I'm a PhD student from the McLean Lab at the University of Southern California. I'm excited to be here today and share our work on single-cell calcium parameter inference. So let me start with the objectives of our work. Let's say that we have data of intracellular calcium response trajectories for all cells in a population and a mathematical model for an intracellular calcium pathway. Our first objective is to determine cell-specific parameter values efficiently for every cell in the population. If we also have the gene expression of all those cells, we want to know if the gene expression would be able to help in learning model parameters. Once we have learned the parameter values, we wish to find out how parameter values inform us about gene expression. The data we used were collected by our collaborators from the Woolman lab at UCLA. The data consists of a joint assay measuring calcium dynamics and gene expression. So for each cell, its calcium response to ATP stimulation was first measured via imaging for 30 minutes. Immediately following this step, the expression level of 336 genes were measured by MERFISH for the exact same cells. We used a system of ordinary differential equations to model the ATP-induced calcium signaling pathway. The equations describe a chain of responses following ATP binding to purinergic receptors on cell membrane. Those responses are the activation of PLC, the production of IP3, the activation of IP3R channel on the surface of the ER, and finally, the release of calcium ions from the ER into the cytoplasm. So here is an overview of our method. We first come up with some ordering of cells, which can either be random or induced by cell-cell similarity in gene expression. For each cell, we smooth its raw trajectory of calcium response. We then sample the parameters of all cells one by one using a Markov chain Monte Carlo method. Once we obtain the inferred parameters, we analyze how well they fit calcium response and their posterior distribution. For each single cell, we assume that the observed calcium response comes from a generative process. The process begins with model parameters drawn from some, the process begins with model parameters drawn from some prior distribution. The value of all model variables is determined by an initial state, the current time, and the model parameters. The observed calcium trajectory is a noisy version of the actual calcium response. For each cell, we sample its posterior PI from a prior pi using NUTS. NUTS is the acronym of no U-turn sampler, which is a Markov chain Monte Carlo sampler. For the first cell, we use an uninformative prior. For any subsequent cell, we derive its prior from the posterior of its predecessor. We see that cells sampled in this way form a cell chain. If the ordering of the cells is random, we call it a random chain. If cell I and cell I+1 are similar in gene expression for all I, then we call the cell chain a similar chain. There are many metrics that we can use to evaluate inferred parameters. To keep things simple, I'm only going to show you log posterior likelihood, which is reported by NUTS, and the trajectory distance, which is the Euclidean distance between input trajectory and trajectory simulated from inferred parameters. Now let me show you the advantage of using cell chain. We sampled 10 cells one by one under 3 different settings. The first one uses a similarity-induced cell chain, which we call similar R1. 500 1-mark iterations are run before actual sampling starts. The other two settings both use the same uninformed prior for each cell, and the number of warm-up iterations was set to 500 and 1,000 respectively. Okay, so here we can see that in general, simulator R1 has better posterior likelihood and needs shorter time to finish sampling. So we conclude that our strategy improve both posterior likelihood and sampling efficiency. Okay. We then sampled another 490 cells for the cell chain similar R1 and plotted the marginal posterior distributions for all cells along the cell chain. While we didn't see anything strange for most parameters, two parameters, B and eta1, caught our attention. Their posterior distribution seemed to be drifting as we go down the cell chain. So it seems that those two parameters were insensitive to cell-to-cell variability. So we were curious what would happen if we set one of those two parameters or both of them to constant values. We ran cell chains on 3 additional models. In reduced 1, eta1 is set to constant. In reduced 2, BE is set to constant. And in reduced 3, both B and eta1 are set to constants. We sampled the same 500 cells as in similar R1. So cells sampled from each model still form a cell chain, a similar chain. Okay, as you can see on the right, we observed no significant change in posterior likelihood or trajectory distance. So we will use reduced 3 for further analysis. Next, we analyzed sample parameter in pairs. We observed competing sources of cell heterogeneity. For example, C0 and eta3 are positively correlated intercellularly, as shown by the top left figure, as well as intracellularly, as shown by the remaining figures in the first row. Epsilon and eta2, on the other hand, are negatively correlated both intercellularly and intracellularly. However, in the case of K-ATP and K-off ATP, the intercellular correlation is positive, whereas intracellular correlations are negative. We also performed an interesting experiment with parameters by perturbing each of them along its marginal distribution. Let's say we're interested in a parameter theta j here. We first pick a few draws in the posterior sample of a cell. For each draw, we replace theta j with an extreme value in the marginal distribution, say the 1 percentile of theta j or 99 percentile of the theta j. We then simulate trajectory using each perturbed draws. Okay, so we saw that calcium response is not sensitive to change in some parameters. Perturbation in other parameters would alter either peak calcium response or steady states. So we have so far shown results from similar chains. So what about random chains? Okay, let's first compare some basic metrics for reduced 3 and to random chains. So as we can see on the right, there's no significant difference in sampling time and the trajectory distance. But this is the whole story. Okay, so now let's look at how gene expression and parameter posteriors are correlated. For each cell, we first computed posterior mean of each parameter for each cell, and then computed Pearson correlation between each gene and each parameter. We ranked gene parameter pairs by absolute Pearson correlation. The top 10 gene parameter pairs from reduced 3 are shown on the right. We can see that some genes are highly correlated with more than one parameter. Meanwhile, some parameters are highly correlated with more than one gene. I'm not going to show you the same table for random chains, but I can tell you that those correlations are lower in general. We then come up with a way to locate cells with similar gene expression in the parameter space. We performed PCA on the posterior sample of a chosen cell, which we called focal cell. We then projected posterior samples of all cells onto the first principal components of the focal cell. So for reduced 3, cells with similar gene expression are closer. This observation can be verified by the distribution of distance between projected samples and the origin of reduced parameter space of the focal cell. For random 2, though, the cells with similar gene expressions are not necessarily closer. At this point, we wondered, can we find out more about parameter space and its relationship to gene expression? So we tried clustering. First, we cluster cells by their gene expression. Then we do a separate round of clustering by the posterior distribution of parameters. This can be done by first computing posterior means of each parameter for all cells and then performing hierarchical clustering on posterior means using word linkage. So what can we learn from clustering? Let's look at how peak calcium response are distributed in each cluster. It is clear that clustering by posterior means on a similar chain can distinguish peak calcium response, and the same can be said for clustering by gene expression. However, clustering by posterior means on a random chain cannot separate cells with different peak calcium response. So we then determine the marker genes for each cell. Each cluster. According to clustering by posterior means on a similar chain and clustering by gene expression, cells with low calcium response were characterized by upregulation of CCDC47 and PP1 family genes. However, clustering by posterior means on a random chain cannot distinguish gene expression differences. So our conclusion here is the posterior distributions from similarity-based cell chains capture the information regarding the transcriptional states of cells. The information captured overlaps with the information that we can directly learn from clustering by gene expression. So that's all for my presentation. I'd like to acknowledge everyone in my lab, our collaborators, and our funding source. And I also like to thank the organizers of this conference for giving me the opportunity to present here today. And I'm now open to questions. Thanks, everyone.
Could you clarify if you used the Murfish data in any spatial way, or was it just to get to expression similarities?
So we didn't use in spatial way. We are just getting like a count matrix.
A count vector.
Yeah.
Okay.
Yeah.
Thanks.
Mm-hmm.